Right, I think we can start. So, what we've already discussed is that the liver does some jobs, and in order for it to do those jobs, it needs these two blood supplies, and those blood supplies give the liver cells the substances that they need to, in order to work on, the materials that they need to carry out the metabolic processes. And as the blood leaves, all those metabolic processes result in a change in the composition of the blood, and so the blood that leaves via the hepatic vein, that then goes to the heart and then to the rest of the body in a more manageable, in a more optimal composition. Okay, so here we have that. So I guess this last bit of the discussion should help us to tie all the things that we discussed at the beginning. We'll come back to that and see how those things are achieved by the cells of the liver. Okay, we're not going to go into too much detail of the metabolic processes, but we are going to see visually how the liver cells achieve all those functions, how they alter the composition of the blood. Okay, so let's begin. So what we said was that you get this blood coming in, right? Now this is the hepatic portal vein. Okay, so a branch of the hepatic portal vein, and remember it's going to have the nutrients from the digestive process. Okay, so we are going to have glucose to deal with, we're going to have amino acids to deal with, we are going to have fatty acids to deal with. We are not going to have oxygen from here, but we're going to have oxygen here. Okay. What else did we say? Potentially, we've got toxins. For example, we could have toxins, for example, hydrogen peroxide, for example, alcohol, and other drug molecules, but these are kind of examples of those things. Okay, so let's remind ourselves of the processes that, or the metabolic functions that the liver carried out, and we'll see how they get done. Okay, so we grouped them in terms of molecules to begin with. So we talked about proteins. All right, we talked about the idea that excess proteins get deaminated, and with that we produce urea. And in relation to the proteins as well, we also get that recomposition of the proportions of amino acids, so transamination. Okay, so those key processes here, we've got deamination, we've got transamination in relation to proteins. In relation to carbohydrates, remember, in relation to carbohydrates, we have glucose homeostasis. Glucose homeostasis, i.e., raising blood levels. If the signals from the rest of the body are, so if there's insulin in the blood, we need to lower the blood glucose levels. If there's glucagon in the blood, we need to increase blood glucose levels. That is also done by the liver cells as the blood flows past. In relation to lipids, okay, we had lipid storage and packaging, so we got lipid metabolism happening here. Right, we also talked about the production of bile, so we'll talk about that. We also said that there is detoxification happening. So there's detoxification going on, and we said that there's a high likelihood that pathogen or the blood arriving at the liver could have pathogens in it because of the food intake from the environment. So we also had pathogen destruction. So we need to see how this microscopic structure is able to do all of those things. Okay, let's begin then. Let's begin with proteins. Okay, so in relation to proteins, what would happen? So we got amino acids from the diet. We've got amino acids from the digestive system arriving. Okay, am I recording? So we got amino acids, right? Now what would happen is the amino acids would, oops, I don't want to use green. The amino acids would arrive, okay, and they would be at a high concentration in the blood. So they would diffuse into the hepatocyte. The hepatocyte would then carry out deamination, producing ammonia. Then following that, we would get the ornithine cycle happening. And as a result of that, the cell would produce urea. Now, urea would then be at a higher concentration here than in the blood. So urea would then diffuse out. And so the blood that leaves, as the blood flows past, amino acids are going in and urea is coming out of the hepatocytes. Okay? So that essentially, as the blood flows past, this is what happens. All the cells in turn are contributing a little bit to that process. Okay? But for explanation purposes, I'll show different cells doing different things. Okay? So that's us dealing with proteins. Also, transamination, as we said, transamination is also a thing. So maybe we get amino acid 1 coming in here. It would diffuse into the cell. Okay, then amino acid 1. Now the hepatocytes are specialized. They're expressing enzymes that are not expressed in other cells. Okay, so as the amino acid 1 comes in, the appropriate metabolic pathways exist within these cells because these cells have certain enzymes that other cells don't. And through these various transamination processes, so let's transamination happens, and it allows us to convert amino acid 1 into amino acid 2. So suddenly now amino acid 2 is at a higher concentration in here than out here, and so amino acid 2 can diffuse out into the blood. All right, and so the blood might come in with a certain composition of amino acids, which, as we've said, it might not be suitable, but by the time the blood gets to around here, the proportion, as we've had all these hepatocytes able to work on the blood and its composition of amino acids through transamination, that by the time the blood gets here, the blood has the appropriate concentrations of amino acids suitable for the protein synthesis happening in our bodies. Okay, so I think we've kind of dealt with what happens to the proteins. Next, let's move on to carbohydrates. And this is going to be a bit interesting because it's not just one story. We can have two situations possible. Okay, so let's follow the story along. Just after a meal, we would have high blood glucose levels. Okay, but we'd also have insulin in the blood as well, released by the beta cells of the pancreas. Okay, and there'll be insulin around, and insulin will be binding to these hepatocyte cells as the blood flows past, and so the hepatocyte cells will be readjusting their metabolic activity to absorb glucose from the blood and convert it into glycogen. So as this glucose flows past, it will diffuse into a hepatocyte as long as insulin is in around. So I'll just put in brackets with insulin present. Okay, so if insulin is there, the glucose will be taken up into the cell. Okay, the glucose will then be converted into insoluble glycogen, and here it will be stored in this hepatocyte. Okay, remember it's happening in multiple places, but we're just kind of visualizing that here right now. So in goes glucose, converted into glycogen, and as the blood flows past, then it has a lower level of glucose than the blood coming in here. Okay, so remember we called that process glycogenesis. Okay, when insulin is around and blood glucose levels are high. However, the opposite situation could also be present. Maybe, or the opposite situation could also happen. Maybe blood glucose levels are low. Okay, and in that case, we wouldn't have insulin in the blood. We would have glucagon. And glucagon, okay, glucagon has the opposite effect on the carbohydrate metabolism of hepatocytes. It stimulates them, it activates them to hydrolyze the stored glycogen back to glucose, mobilizes glucose, glucose then enters the blood, and blood glucose levels rise. So we could say, in the presence of glucagon then, what would happen is the opposite to this, which is that glycogen will be hydrolyzed to glucose and increasing the glucose concentration in the hepatocyte, and therefore causing glucose to diffuse into the blood, thereby raising the blood glucose levels when it's the time to do that. So I hope we can appreciate these wouldn't be happening at the same time, they would be happening. So these processes are dependent on the glycemic situation of the body, okay, or of the blood. Okay, so that's kind of carbohydrate metabolism dealt with there. And I mean, this is just to illustrate how this might happen. So as the blood flows past, you know, if glucagon's around, glucose would be coming out of these hepatocytes into the blood, and so the blood leaving through the central vein there, that would have higher blood glucose levels in the presence of glucagon. Okay, so we've kind of dealt with proteins there, we've dealt with carbohydrate metabolism. Let's talk about lipids. Okay, so in the situation where, you know, you've just had a meal, there's high levels of fatty acids in the blood, glycerol also, this would then flow past to the cells. What do these hepatocytes do with that? Okay, so what would happen is that the fatty acids would enter a hepatocyte or hepatocytes, okay, glycerol also, glycerol would enter, and here they would be recombined through condensation reactions. They'll be recombined and they would form triglycerides. 3 fatty acid chains and glycerol, remember, the energy storage form of lipids. And these triglycerides, they are not really stored in the liver, or they shouldn't be. What happens is they get packaged up into these structures called lipoproteins, which are structures that can kind of bulk transport lipids from one place in the body to another. Okay, so these lipoproteins then enter the blood, and they will then be transported to the fat stores of the body, where they will be absorbed into the fat storage cells, i.e. adipose tissue, etc. Okay, so off those lipoproteins would go in the blood. So blood would come in with fatty acids and glycerol, but it would leave with lipoproteins. Okay, next, what else could happen? So we've discussed lipid metabolism. Next, we've got bile production. Now, other hepatocytes will be carrying out the production of bile. This, what we've discussed here, this structure, this area here, this is called a sinusoid or a sinusoid, however you want to call it. But here, in this little channel where bile is being produced, or the bile salts are being released. Okay, so we got all of these cells releasing, you know, the components of bile into this little space here. This is called a bile canaliculus. Okay, like a canal. All right, so we got bile being released here, and this collects and moves in the opposite direction. Bile here, and off it goes and taken away via the bile duct to the gallbladder. Okay, and then eventually used in the digestion of lipids later. All right, so we have now talked about bile production. So other hepatocytes right here secreting bile into this area, and off it goes. What else do we have? We have detoxification. Okay, detoxification. So as this blood would flow. Now, there's no. It's not necessary that the toxins are also just in the hepatic portal vein. Toxins could just as easily be present here. Okay, so just for space purposes, let's say that we have some hydrogen peroxide. Okay. A molecule of hydrogen peroxide just enters in through here. Now, what would happen to hydrogen peroxide? I think you can tell where this is going by now, but just to complete our story, a stickler for completeness, okay? But hydrogen peroxide would be here. It would diffuse into the hepatocyte. The hepatocyte would be expressing the enzyme catalase, okay? So it would have catalase. So the hydrogen peroxide, the catalase would catalyze the breakdown of hydrogen peroxide to oxygen and water, half an oxygen molecule and water, okay? And those two could then diffuse out of the hepatocyte. Nice, and that's really all we're talking about. Similarly, if there are other toxins such as alcohol, you know, the textbooks describe the metabolic pathway through which alcohol is converted into acetyl units that can be used to make fatty acids or used in respiration. Okay, but similarly, those other toxins could go into a cell and then metabolized like that. Okay, so we have an example of detoxification happening, and other detoxification reactions would happen in a similar way. Okay, so we've talked detoxification. Last one on our list is pathogen destruction. Pathogen destruction. So Kupffer cells are these macrophages that patrol the sinusoids. Okay, let's draw one here, crawling along. Okay. Yes. Okay, so these Kupffer cells are macrophages that patrol these sinusoids. And essentially what they are doing is if they do come across any pathogens, what they will do is carry out phagocytosis, engulf those pathogens and carry out phagocytosis to digest or break those pathogens down so that the blood leaving has fewer pathogens in it than what arrived. And that is essentially the destruction of pathogens. So I hope what we have done here is kind of justified the structure of the liver, kind of from a macroscopic point of view, looking at why it needs certain blood supplies. I hope we have tried to marry that up with all the functions that the liver produces, and then we've seen on a kind of microscopic scale the sinusoid structure, the lobule structure is carrying out all the functions that we have discussed on a microscopic scale that is simply multiplied many times to give you the large liver organ, but all of those lobules carrying out essentially all the functions of the liver just on a small scale, which is then multiplied up to increase the surface area and rate at which the liver can carry out all those processes, so that the blood that leaves the liver is appropriately recompositioned to make it more suitable for the functioning organism. Okay, I hope that that has been the story that I have told you guys. Any questions, do ask in the comments. As always, I really appreciate that you guys are watching these long videos. An uninteresting person like me who is discussing these things. Well done to you for persisting and working hard, okay? Going through that pain and anguish to get to the other side, which is undoubted success, guys, if you keep working, okay? So keep going, keep working. The misery will be followed by success, okay? If there is enough misery, there will be enough success. All right, so guys, thank you for paying attention. If there's any questions in relation to any of this, please do ask. I'm not the world's expert on any of these things. It's just that I think by kind of breaking certain things down, by simplifying them to a point where we can understand it, from that point we can then build up towards a higher level of detail, complication, but we first need to break it down to its simplest components and then build it up from there. All right, guys, I hope you have found this useful. This has been the liver. Keep going, guys. Power.